My name is Madeline Lofton, and I am the Director for Educator Professional Learning at the Hudson Alpha Institute for Biotechnology in Huntsville, Alabama. Hudson Alpha is a nonprofit biotechnology research institute that has a threefold mission. Our primary mission is to use the power of genomic research to solve big picture problems, investigating the underlying genetic and genomic causes of things like heart disease and disorders of the mind, using DNA sequencing to diagnose rare disorders, working on plants using the same DNA sequencing technology to identify important traits in agriculturally significant plants. Hudson Alpha also focuses on economic development. How do you take the discoveries that researchers make and move them into the business world, into a clinic, doctor's office, hospital? The third leg is educational outreach. If we are going to use the power of genomic research to solve problems, then we're going to need to grow and train the next generation of researchers, the next generation of the biotech workforce. In the educational outreach office, we develop materials that support teachers in teaching genetics and genomics content. We lead student experiences. We engage with the public and patients, all with a goal of building a more genomically literate society. Hudson Alpha kits are developed with the end user in mind, the student. They're designed to be used in life science classrooms, but they have a very deliberate focus on the career connections. We want students to understand that being engaged in science or studying in science is more than becoming a doctor or a nurse or a research chemist. There's a deliberate focus on how does the biology content relate to potential careers. The other thing that makes Hudson Alpha kits unique is the team that works together to design them. Our educational outreach team is made up of educators, genetic counselors, and research scientists so that we make sure we get the science right and also that we have clearly communicated those concepts to students. Many teachers have a favorite karyotyping activity. Unfortunately, many of those require cutting X's out, and so you wind up with paper all over the floor. Disorder detectives uses something completely different. We're going to use chromosome clings on a game board so that students get an experience that's more similar to actual karyotyping. These chromosome clings are going to be matched up with the ideograms that are already on the board. In addition, Disorder Detectives provides teachers an opportunity to look at the limitations of karyotyping. However, it is still an amazing tool to help students understand how chromosomes behave during cell division and to diagnose some common genetic disorders. Students have significant misconceptions when it comes to genetics and inheritance pattern. And many of those actually stem from a misunderstanding of the way chromosomes move during cell division. Hudson Alpha's Chromosoc kit is a way to put chromosomes in students' hands. Socks come in pairs, chromosomes come in pairs. So we're going to use the socks in students' hands to allow them to move the sock chromosomes the way that chromosomes behave during cell division. The best part about all of the Chromosocs kits is that it allows the teacher to see what the student is thinking. Having students move socks enables the teacher to identify and address misconceptions early in learning about this process. The Modeling Mendel's Laws kit takes that a step further. You can use socks to teach Mendelian inheritance patterns. So we're anchoring Mendelian inheritance patterns in the biology of chromosome movement during meiosis and extending it even further and using that to illustrate how Punnett squares work using the socks before we start putting Punnett squares on the whiteboard. Genes and consequences puts students into a modern biological database, the same database that researchers use. Students are going to use this to get comfortable, to ground themselves in the central dogma. The Genes and Consequences kit uses an older sequencing technology, Sanger sequencing, and electropherograms that are produced by that sequencing to really reinforce nucleotide bonding, to keep up with A bonds to T and C bonds to G. Genes and Consequences then takes students through the process all the way down to a sequence of amino acids. And then students are presented with a patient card, and they analyze that patient sequence as well. What students discover when they blast both the typical and the patient sequence is that the hits will be in the same gene. And this enables teachers to get at that misconception that a patient has a gene, say, for cystic fibrosis, when we all have the cystic fibrosis membrane transport protein. It matters which variant of that protein you have and how that impacts the amino acid sequence. Students are familiar with cancer. 1 out of every 3 American women and 1 out of every 2 American men will be impacted with cancer at some point in their life. Collecting cancer-causing changes shows how over many cycles of cell division that cells accumulate mutations, and that some of these mutations can lead a cell to become more cancer-like. The kit uses beads to illustrate the outcomes of cell division, and it ties random mutation events to checkpoints in the cell cycle that would control the cell's progression toward cancer. This shows students that your genes are not determinate, that they are not fate, that there are other external factors that may impact the likelihood of developing cancer. And students can see that over the course of multiple cell divisions using beads and dice. HMPCC is for the biology teacher the complete package. Set in a storyline of an inherited form of colon cancer, students learn how to construct pedigrees and how to use information from that pedigree to predict who in the family might be at an increased risk for colon cancer and who might not. Students then get DNA samples from family members and actually run gel electrophoresis to look for the presence of this DNA variant that increases risk for colon cancer. And then students talk about the potential ethical considerations of population screening. They take on the role of a genetic counselor and use those gel electrophoresis test results to talk to family members about earlier cancer screening or potential lifestyle changes to reduce their risk for colon cancer. Gaudy Goldfish uses the concept of fancy goldfish and monetary drivers to engage younger students in thinking about inheritance patterns and genetics concepts. Students are challenged to discover the role of genes as they relate to visible traits in fancy goldfish. Things such as the length of the goldfish tail, or how tall a dorsal fin is, or the coloration of the goldfish. Students make a series of breeding decisions so that they discover the inheritance patterns for these visible traits. And then they're challenged to use what they learn about inheritance patterns to build a breeding plan to develop the fanciest goldfish that they can come up with, that they'll be able to sell for the most money at the pet stores. And one of my favorite features is that it prompts students to build conceptual understandings of these inheritance patterns, like dominant and recessive and even codominant, before they necessarily have the vocabulary. For example, in piloting this kit in classrooms, one of the things that I was able to see is a student explain that a particular trait they were looking at, you could only get that trait if there were none of the other version of the trait there, to which the teacher replied, yes, you're absolutely right. It will only show up if both parents have that trait. The student was able to build that conceptual understanding well before they had the vocabulary. We would like to see every biology student across the country or across the world using Hudson Alpha Kit, and those students having those aha moments. And one of the best ways that we could expand our reach is by partnering with the leader in life science education, Carolina Biological. I think the materials that we are developing since the partnership with Carolina are stronger, are more effective in the classroom. We're more conscious of how our materials are chosen and how those are going to be used in the classroom. Partnering with the leader in life science kit materials and resources was a really easy choice for Hudson Alpha.